Hi everyone, welcome to our channel. Today, we're diving into the recent tragic deaths linked to gene therapy for Duchenne Muscular Dystrophy, DMD. This isn't just a story about cutting-edge medicine. It's a profound lesson in safety and a call to learn from experience. Duchenne Muscular Dystrophy, commonly known as DMD, is an X-linked recessive genetic disorder. This means it primarily affects males, while females are typically carriers. Globally, about 1 in every 5,000 boys is affected. The culprit is a defective gene located on the X chromosome, specifically in the XP21.1 to XP21.3 region. This gene codes for a protein called dystrophin, which is critical for maintaining the structural integrity of muscle cells. Think of dystrophin as a scaffold that stabilizes the membrane of skeletal muscle cells. In DMD patients, a genetic defect leads to the absence of dystrophin, making muscle cell membranes extremely fragile, like a wall without reinforcement. Without this protective layer, the membranes are easily damaged by external stress. When the membrane breaks, cellular contents leak out, triggering the immune system to recognize these as danger signals. This activates an innate immune response, sparking localized inflammation. This inflammatory response further damages muscle tissue, trapping muscle fibers in a cycle of repeated destruction and incomplete repair. Over time, chronic inflammation causes muscle tissue to be replaced by fibrotic tissue, making muscles stiff, weak, and unable to contract effectively. Additionally, membrane damage leads to an overload of calcium ions inside cells and heightened oxidative stress, both of which further impair muscle cell regeneration, causing cells that could have recovered to lose vitality. DMD patients begin experiencing progressive muscle weakness from early childhood, eventually losing the ability to walk. The condition can also lead to cardiomyopathy and respiratory failure. On average, patients face a life expectancy in their late teens to early 20s. At its core, DMD is driven by a cascade of issues stemming from the dystrophin gene defect. This is why gene therapy is so critical for DMD. Traditional treatments can only slow disease progression, but they cannot address the root genetic cause or restore dystrophin production. Adeno-associated virus, AAV gene therapy, however, delivers a functional microdystrophin gene to muscle cells, restoring dystrophin-like protein expression, improving muscle function, and significantly slowing disease progression. This approach has the potential to transform DMD from a fatal condition into a manageable one. Sarepta Therapeutics therapy uses AAV vectors as a delivery tool to transport the microdystrophin gene into muscle cells. AAV vectors are considered non-pathogenic, non-integrating, and have a strong affinity for muscle tissue. In 2019, Sarepta acquired key AAV gene therapy projects and technologies, achieving promising early results in preclinical and initial clinical trials. Between 2023 and 2024, the EMBARK Phase 3 trial was conducted globally, a placebo-controlled study enrolling ambulatory DMD patients to confirm clinical benefits. In June 2023, the FDA granted accelerated approval for ELEVIDYS, specifically for 4 to 5-year-old DMD patients. In January 2025, EMBARK reported positive results, demonstrating sustained treatment benefits and meaningful disease stabilization. However, earlier this year, devastating news emerged. 2 non-ambulatory teenage DMD patients died from acute liver failure after receiving Sarepta Therapeutics' ELEVIDYS gene therapy. A third death followed. A 51-year-old patient in a limb-girdle muscular dystrophy trial, who also used the same AAVrh74 vector. These incidents quickly drew intense scrutiny and intervention from the U.S. Food and Drug Administration, FDA. On July 18, 2025, the FDA requested that Sarepta voluntarily halt all ELEVIDYS shipments. Additionally, the FDA imposed a clinical hold on the limb-girdle muscular dystrophy trial using the same vector and revoked Sarepta's AAVrh74 platform technology designation. Initially, Sarepta resisted halting treatment for non-ambulatory DMD patients, arguing that no new safety signals had emerged in this group. However, by July 22nd, Sarepta partially complied, voluntarily pausing shipments to non-ambulatory patients and agreeing to update ELEVIDYS label with a box warning about liver damage. They are also in ongoing discussions with the FDA to maintain treatment access for ambulatory patients. If safety concerns outweigh benefits, the FDA may fully withdraw Levity's approval. The duration of the current pause or additional trials remains unclear, potentially extending into the future. The review process could last until the end of this year or longer. The outcome will not only shape the future of this therapy, but also set a regulatory precedent for gene therapies broadly. These events have significantly impacted Sarepta's stock and operations. Since March, Sarepta's stock has plummeted by 86%. In response, the company announced a major restructuring in July 2025, laying off 500 employees, 36% of the workforce, and implementing a $400 million cost-saving plan. Sarepta also faces a looming $1.1 billion debt due in 2027. For patients, the impact is profound. Many DMD families who secured insurance and scheduled treatments now face delays, compounding emotional and logistical challenges. Let's dig into the cause of these deaths. Liver toxicity. The two teenagers died from acute liver failure following the Levity's treatment. Patients with advanced disease may be at higher risk, as non-ambulatory patients typically have more progressed conditions. The third death, in the limb-girdle muscular dystrophy trial, may also be linked to systemic immune responses and liver inflammation. Liver toxicity is likely driven by an immune response triggered by the AAV vector. The Levity's uses the AAVrh74 vector, a modified adeno-associated virus effective at delivering the microdystrophin gene. However, high doses can provoke severe immune reactions, including immune activation against liver cells, inflammation, or T-cell-mediated liver toxicity, and in rare cases, even a cytokine storm. Patients with advanced disease often have compromised heart, respiratory, and liver reserves, making them less resilient to treatment-induced systemic stress. The fact that early-stage, ambulatory, and late-stage patients receive the same dose may have overwhelmed certain systems. These incidents offer critical lessons. First, gene therapy carries risks. While the idea of a one-time fix is appealing, even conditionally approved high-dose gene therapies remain experimental. Patients should inquire about long-term safety data, not just short-term improvements. Second, understand FDA approvals. The Levity's received accelerated approval based on early data from ambulatory children, but it wasn't extensively tested in non-ambulatory teens or adults. When considering treatment, ask critical questions. Has this drug been tested in people like me? Age, disease stage, organ function? And is this a full or conditional approval? Third, prioritize transparency and shared decision-making. Sarepta initially didn't disclose the deaths publicly. It was the FDA that mandated transparency. As patients and families, we must demand full risk disclosure, clear monitoring plans, and access to FDA correspondence or company risk communications. Fourth, weigh cost against risk. The Levity's cost approximately $3.22 million per treatment. But remember, a high price doesn't guarantee safety. Expensive therapies can still carry significant risks, often backed by limited real-world data. In summary, this tragedy is a wake-up call. It underscores that even breakthrough therapies can pose life-threatening risks, especially in vulnerable populations. We must ask critical questions and not hesitate to discuss risks and benefits with healthcare providers. Understand the difference between conditional and full FDA approvals. Demand oversight, insisting on rigorous safety monitoring and transparent communication. Above all, make informed decisions, especially for gene therapies still under active investigation. That's all for today. Remember, staying informed is your best protection when considering new medical treatments. If you found this content helpful, don't forget to like, subscribe, and hit the notification bell. Thanks for watching, and we'll see you in the next one.